Hello everyone. I am Srashti Maheshwari, Assistant Professor in IIMT College of Pharmacy, Greater Noida. Today, we are going to discuss about pharmaceutical validation. So the content which we are going to cover is definition of validation, validation of specific doses form, types of validation, cleaning validation, equipment validation, and the process flow chart for validation, analytical method validation, as well as the parameters involved in analytical method validation. So, what is validation? Validation is a documented program which provides a high degree of assurance that the specific process will consistently produce a product meeting its predetermined specification standards and quality attributes. Validation is the process of evaluating products and analytical method to ensure compliance with product or cleaning methods requirement. So the validation is the mean of catering amorphous benefits to even more than the acceptable quality level, which in the global standard scale. Leading importance to validation is increased profound in recent years. The validation is basically the art of designing and practicing the designing steps alongside with the documentation. Basically, validation is a all documented program. So, as per the ICH, validation of analytical method is a process by which it is established by laboratory studies which the performance features of the methods meet the requirements of the predetermined standards. As per the FDA, validation is establishing documented evidence which provide a high degree of assurance that a specific process will produce a product meeting its predetermined standards and quality attributes. So the next point is validation of specific doses form. So specific doses forms involves tablets, capsule, liquid formulations, powders, and any other form. It is crucial to assure their safety, efficacy, potency, consistency. There may be various skill steps involved in the validation for a specific doses form. First is formulation development. The first step is to develop the formulation that meets the required specification for the doses form. This includes selecting appropriate ingredient, determining their proportion or compositions, optimizing the manufacturing process. Then next point is analytical method development. Analytical method development basically validating a doses form required reliable analytical method to assess its quality attribute. This involves developing and validating analytical methods for testing parameters such as purity, potency, stability, dissolution rate, disintegration rate, etc. Next is process validation. Process validation assure that the manufacturing process consistently produce doses form of the desired quality. It involves establishing process parameter, conducting process qualification studies, and monitoring process performance over time. Next is stability study. Stability study assess the shelf life, duration, and the stability of the product. Form of the doses form under various storage conditions. These studies helps determining appropriate storage conditions, shelf life, expiry date, duration, and packaging requirements. Next is quality control testing. Quality control testing involves routine testing of finished dosage form to ensure that they meet predefined specification for identity standards, purity, quality. This includes testing for physical characteristics such as appearance, uniformity, texture, as well as the microbial and the chemical attributes. Next is documentation and the reporting. So validation process throughout the validation, its detailed documentation is maintained to provide evidence of the compliance with regulatory requirement and internal quality standards. These documentation includes validation protocols, reports, batch records, master records, and standard operating procedures. Next point is regulatory compliance. So regulatory compliance is set for by health authorities such as FDA, United States Food and Drug Administration in America, or EMA, European Medicine Agency in Europe. This involves submitting validation data as per the regulatory filing for marketing approval. By the systematic approach to validation, by following the systematic approach to the validation, pharmaceutical companies can assure the safety, efficacy, as well as the quality of the product, ultimately benefit the patient and the healthcare providers. Next is types of validation. There are major four types of validation. First is process validation. Next is cleaning validation. Third is equipment validation. And last one is validation of analytical methods. So, what is process validation? Process validation means of assuring and providing documentary and evidence that processes are capable of repeatedly and reliably produce a finished product of the required quality. Prerequisites of the process validation: qualification of the manufacturing equipment and the instrument, as well as the formulation. This involves compatibility of API, recipient of finished product, and packaging material. Critical services involve water supply, power supply, air, nitrogen, equipment cleaning, and motivation of personnel. Types of process validation. There are basically four types of process validation: prospective validation, concurrent validation, retrospective validation, and the process revalidation. So the first point is prospective validation. Prospective validation basically involves the development stage. It carried out during the development stage by means the risk analysis of the production process, which is broken down into the individual steps. These are then evaluated on the basis of past experience to determine whether they might lead a critical situation or not. Expected the process validation be completed prior to the release of the product for the sale. It is a basically initial stage of the formulation development and the process development. It's involved process sampling plans, designing of batch record, defining raw material specification, transfer of technology from scale up, the batch scale, to commercial size batches, environmental controls. So the prospective validation is considered acceptable than 3 consecutive batches within the final accepted parameters that assure product of the desired quality both constitute a proper validation of the process. Next is concurrent validation. Concurrent validation is used for the particular batch currently being produced. No past experience is involved here. So it is used on the batches currently being produced for the establishing documented evidence that the facilities and the process do what they claim to do. For example, a previous validated process is being transferred to the third party contract manufacturer or the another site. The number of batches produced are limiting. Process with low production volume per batch and the market demands. The product is a different strain of the previous validated product with the same ratio of API and the excipients. Retrospective validation. So retrospective validation is the examination of past experience. It involves the examination of past experience of the production on the assumption that composition, procedure, equipment remain unchanged. Such experience and the result of in process and the final quality control tested are the evaluated. Recorded difficulties and failure in production are analyzed to determine the limit of process parameter. So the main point is retrospective validation is obviously not quality assurance measure itself. It should never be applied to the new process or the product. Basically, it is a examination or the analysis of past experience. Next is process revalidation. Revalidation means repeating. So the process revalidation is required when there is a change in any critical process parameters, formulations, such as equipments, packaging material, raw material, packaging components, and the formulation. For example, change in raw material. Change in raw material involve physical properties such as density, viscosity, particle size, distribution, or change in source of active raw material manufacturers. Next is change in packaging material. Change in packaging material involve primary containers, glass to plastic or plastic to glass, closure system. Change in process. Change in process involve mixing time, drying temperature, batch size, change in equipments, and the change in plant facilities. Next is cleaning validation. Cleaning validation is all about contamination. Means cleaning validation ensure that there is no contamination or cross-contamination in a multi-product manufacturing plant and also prevent microbial or any chemical contamination. Objective of cleaning validation is to minimize cross-contamination, to determine cleaning process efficacy. Next is source of contamination. So the cross-contamination product of one product into another. Product contamination by unwanted substance, foreign materials, microbial contamination. So the factors influencing cleaning validation: product, equipments, facilities, cleaning method, cleaning agents, sampling, testing, limit, and acceptance limit. They all play a major role in the cleaning validation. Next is equipment validation. Equipment validation provide a higher level of documented evidence that the equipment and the process conform to a standards. Equipment validation are four types: design qualification, installation qualification, operational qualification, and the performance qualification. Design qualification is a very first step. Basically, it's done before the purchasing. After that, installation qualification is done for the new equipment or the updation of the last equipment or the previous equipment. Then operational qualification, how it is work according to the standards or not. Then the performance, how it is performed properly. Is there any issue? It's record on the daily and weekly and monthly basis. So the process flow chart of the qualifications. First is design qualification. Design qualification basically done before the purchasing new instrument. Then installation qualification. Installation qualification is documented installation process or the installation report of a new and the existing equipment. Then operational qualification done after the installation or after the major change in the instrument, such as after repairing, updation, regular intervals, risk based. And then performance qualification done after instrument issued on daily basis, weekly basis, monthly basis. Next is analytical method validation. So a method validation must prove that the analytical method used for the specific test is suitable for which it is carried out. Methods should be validated when they are to be established on routine use. Method is to be changed due to the change in condition. Whenever the equivalence between new method and the standards are demonstrated. Now the parameters of method validation. The parameters involve first accuracy. Accuracy refer as the closeness of agreement between observed value and the true value or the reference value. Second point is precision. Precision is closeness of values obtained from the series of tests. After the various tests, the closeness has been measured. Then repeatability. Repeatability is established when the same sample is examined repeatedly by the same analyst during the analytical method within the same lab, same instrument, and performed within a short period of time. Then fourth point is intermediate precision. Intermediate precision is when the test is repeated on the different day by different person under the using the different instrument, different conditions within the same lab. Then the variation is expressed in terms of intermediate precision. Then reproducibility. Reproducibility is a standardized method. The test is carried out in a different laboratory using the same method. The precision between laboratory is known as reproducibility if there is certain change or none has been recorded. Then specificity. Specificity is the ability of test method to measure the analysis explicitly in the presence of other components. Next is linearity. Linearity is referred to its ability to measure a specific component using a range. Detection limit is basically a lowest lowest detectable concentration of an analytes in a sample. Quantitation limit, the lowest measurable concentration of an analyte in a sample. Next is robustness. Robustness is the ability of a test method to resist the variation in method parameter. Robustness is basically done for the small variation. Then the range. Range is interval between upper and lower concentration of analysis. So the summary is validation. Validation is basically a documented program. So quality and the hence quality play a major role in the pharmaceutical industry. It's any industry and its product manufacture. Multiple views on obtaining such quality are the current interested in the pharmaceutical industry. At last, validation and the quality assurance will go hand in hand and assure the quality and the product throughout the process, throughout the formulation. Thank you.